Started, I wanted to start with this reminder about the writing assignment. I talked about it yesterday in my discussion. I think Dr. Drury mentioned in her discussion, but just to be clear, also the discussion was not very well attended yesterday. You have a writing assignment. The goal of it is to have you think about a news article that talks about genetics and then make an argument. You agree with their conclusions, you disagree, why. You have to find three things: a news article that cites the primary literature. Primary literature is a scientific study, so that's the second thing, and then the third scientific study that's going to support your argument. So there's a whole module for this. There's a whole module for this. So that's what you see here, and I updated this example because it was broken yesterday, so now it's a PDF. So this is an example article from the New York Times. So I think the question is, how can we tell that it's citing primary literature? It should be either at the end of the article or for the New York Times or often embedded. So you can read about prenatal testing and how people relied on it, but now it's showing it's not actually very accurate. The formatting on this is really weird because I had to print it as a PDF. If you go here, there's a bunch of citations. And then it takes you to a journal, right? Like a scientific study. So this is the Journal of Fetal Medicine. So they did a study, SNP, so these are mutations, non-invasive prenatal testing. So this was a study done by scientists, and then the New York Times made an argument based on the study. You have to make an argument based on these sources and one other source that you find. Don't use this article, find your own article. If you want me or Dr. Drury to check your article, that's fine. You will have to post your article April 4th, and that'll be a checkpoint as well. Any questions on this? Okay. I'm going to switch over. Okay, so today we're transitioning to think about gene regulation. The ultimate goal is to get at the genetics behind gene regulation. So we've seen gene regulation in terms of like this, transcription, translation. Well, what's the goal of that, right? Your cell is making proteins to do function. How is that controlled at the genetic level? So we're going to start with bacteria. Bacteria are really quirky in how they organize genes and regulatory regions. The reason we're starting there is because it's a simple system that we build intuition and then move forward. So we're going to spend a lot of time on these weird partially diploid systems because they're the closest thing in bacteria that approximate eukaryotic gene regulation. Eukaryotic gene regulation is much more complex, the genetics is much more complex. So we're going to start with bacteria, but then try to generalize it. So today will be an overview. Next Tuesday, we'll focus heavily on the last operon and the trip operon. Bacteria, and then on next Thursday, we'll transition to eukaryotic gene regulation. So gene regulation is important for all organisms, and we need three things for gene regulation to occur. So we've already seen these in a little bit in this context. Now we're going to do genetics. So does anyone remember, or just you can yell one of them and put in the chat? If you're trying to regulate a gene, what are the three pieces you might need? I only have like 20 slides, so we can do these awkward silences for like a minute. We're good today. So RNA, yes, so that's a template. So where is the RNA going to come from?
The DNA.
The gene. We need the gene that we care about, that we're regulating. So there's going to be two types of genes. The first one is the structural gene. That's the gene that's like doing the work, right? by work, I mean whatever enzymatic activity in the cell we care about. Yep, we need a promoter. So I would classify that under a regulatory element. One other thing. What's going to connect this regulatory element to the transcription of the structural gene? Yeah, so if someone couldn't have a binder, yeah, I think what you're saying, there's going to be a physical interaction between things. What's going to physically interact? Yeah, we're getting a lot of good answers. RNA polymerase, amino acid, that's downstream. That's like after this gene's been. The receptor protein, that was on the other slide. That's a good idea, right? That's something that's interacting with the environment. I'm thinking of one key thing. A binding protein, yeah. So I think we're going kind of in the right direction. We need a protein that's going to bind to the regulatory element of the structural gene. So that's a regulatory protein, or a regulatory gene. This gene will have its own promoter, but its effect that we care about is on the promoter of the structural gene. So this is going to encode a product that interacts with the structural gene. So it's not necessarily catalyzing a reaction in the cell. Its purpose is to regulate the expression of another gene. And then we have the regulatory element. So these often are not translated or transcribed depending on what type we're talking about. So when I say expression here, that's synonymous with transcription. So transcription is the process going from RNA to DNA. But when we're talking about genetics, we often refer to it as the regulation of genetic expression. So if you hear me saying those things, I've kind of tried to switch my language now away from transcription towards expression. So we're regulating gene expression. You can think of it as transcription. The reason I don't like transcription is transcription is due to polymerase. This regulation is happening prior to polymerase. It's happening at the promoter and regulatory sequence level. But that helps you to make that connection. That's fine. Before we move on, quick heads up. The top half slide, the top half questions are kind of different. They're more like concept checks rather than working problems, because we're not going to work problems till next week. So they might, we'll just see how they go. Okay. So what's the consequence? We touched base on this too. What's the consequences of being able to regulate your genes? Or why is it necessary for a cell to do this? Any ideas? Typing in the chat's fine. I think it's more efficient if you just yell at me, but that's fine too. Yeah, so you can use genes only when it's needed. So I would say express a gene at a specific time. So you can make an argument about efficiency. But what kind of conditions are necessary to just regulate the gene at a specific time? I think there's two main ones. So you can share your ideas too. So we saw an example of this last time through development or different tissues. A liver is not going to express the same genes as heart cells, muscle cells. Bacteria don't have that type of organization. So what is a bacteria? Why does it need to regulate gene expression? This would be a cell differentiation. What about in a single-celled organism? Yeah, homeostasis. So I would say, I would rephrase that in responding to your environment. Extracellular. You're a bacteria hanging out, now you're in a saline environment, or now you're in the gut. You're going to change gene expression in response to the extracellular environment to maintain homeostasis intracellular. Right? So those are the kind of the consequences and the reasons for differential gene expression. Tissue differentiation or cell differentiation and responding to external stimuli. Right? Even in like complex eukaryotic systems, right? Neurons might undergo gene expression change in response to a stimulus. So it's happening in lots of different contexts, but it's all happening due to the genetic control of regulation. And so it can happen at a couple different stages. We've seen this a little bit. It can happen at the structural changes to the DNA, splicing, processing. We talked about those as kind of like de facto processes, your cap, your tail, your splice. But that actually has really important implications for gene expression and genetics. You can regulate proteins in different ways of modifications. But we're going to focus mostly on kind of pre-transcriptional. So whether a gene is on, close, or off. So here's our first top-hat question. Why is transcriptional regulation, so stopping or starting transcription, an important thing to regulate? So we're getting some interesting answers here. I think we already touched on it, but I'll talk about some of these other ones. There's a couple things in terms of mutation. And it doesn't have to be the people who answered this, but what is the mutation? Is mutation going to play? So the question is, why is transcription a good way to regulate the level of a gene? Is mutation going to play a role in that? Maybe, maybe not. I think mutations are going to be important for genetics. You can't have changes without differential gene expression without a different DNA sequence. But mutations are only happening in DNA replication. Right? So I'm not sure what those comments meant, but I think it's a good idea. But replication is where mutations occur. Transcription might be affected by previous mutations, but it's not going to cause mutations. So yeah, some of the things we've already talked about, just cell differentiation. But I think the answer that I prefer is cellular efficiency. So we already touched on this a little bit. Tissue differentiation is a consequence and can encapsulate cellular efficiency, right? You don't want to make products that you don't need, but actually that is a different problem, right? If you're transcribing and expressing genes you don't need, you actually don't get differentiation. So I think the best answer here is thinking about cellular efficiency. It's the first step, right? Why spend all the energy modifying, splicing proteins when you can just turn off and on gene expression kind of at this way? So again, these questions are more of like concept checks to make sure we're on the same page before going forward. So this one's a multiple choice question. So what's the difference between a structural gene and a regulatory gene, a regulated gene? not, does this question not exist? Say that again?
It wasn't very helpful.
Okay. I don't think this question exists. Oh, there it is, it's out of order. Okay, so it should be open. So we've defined these things really quickly. Let's stop and think about them before we proceed. Because if we're not on the same page here, what's coming next might not make as much sense. So this is not worded the best, I recognize that. It's from the book, but I think the book's trying to make a really specific point with this question. So currently there's like even split between C and D. Does anyone want to defend either C or D?
I chose D because in C it says that proteins function in the structure of the cell, but don't they function outside of the cell also?
Sure, there's many ways that you can kind of do an elimination. So that's eliminating C.
Yeah.
Okay, so there's a comment in the chat, a regulator gene helps expression, structural genes of the operon for the book. And then there's a question, I think, do they both encode proteins? Yes, so that's what I didn't like about the wording of this one. So if you read D, structural genes encode proteins, regulator genes control transcription. It is a protein though, so yeah, that's what I don't like about this question. But I think the method of elimination that you were talking about, there's even another one is important. So A, we can eliminate that one, right? Structural genes are not transcribed, or regulator, or they are regulator genes or not. How would they do work if they're not a protein? So that can rule out that one. The structure, you don't know about those. I mean, that's not where it gets its name. So back to C and D, which is what the majority of you are answering. I think this is the key part that I can rule out C. Regulator genes are not doing metabolism. They're not a catalyst. They are interacting with DNA. So I think that's what someone put in the chat, right? It's binding to DNA at the operator of an operon. It's not going to do a reaction. So that can rule out C, so the best answer here is C. Any questions on that? They're both proteins. So that's why these questions not the best. Okay, so now we're going to talk about operons. So operons are very bacteria specific. They don't occur in eukaryotic cells. It's a very specific way of organizing the gene that made them, or a set of genes, and that's what made them really easy to study in early genetic studies of gene expression. So we've heard some of these terms, people have talked about them, we've talked about them. You have kind of two pieces, the structural genes and the regulator genes, but it's only the structural genes that are part of the operon. And bacteria, they're like stacked right on top of each other. They have a shared promoter. That's not the case in eukaryotic genes. Eukaryotic genes have usually a single promoter for a single gene. They might have shared enhancers, but it's kind of a one-to-one relationship in eukaryotes, so it's not the same in bacteria. Bacteria are really sufficient in their genome organization. All the genes, or most of the genes, are going to be important for a specific metabolism or clustered together. So these would be the structural genes. They need to be turned on. They need a promoter. The promoter is where RNA polymerase is going to come on, where the regulatory protein is coming on, is at the, or binding, excuse me, is at the operator. Sometimes in an operon, the operator can overlap with the beginning of the structural gene. So keep that in mind, it doesn't always have to be like this unique spacing DNA sequence. So all together, this is the operon. So the operator is where your regulatory gene is going to come in. The promoter is where RNA polymerase would come in. We need something to regulate this operon. So then we have a promoter. And the regulatory gene. So these two systems are physically separated in the bacterial genome. So that is kind of hopefully foreshadowing some really interesting genetic consequences. If there's mutations in the operon, it's going to have very different consequences than mutations in the regulatory gene because of how they interact. This regulatory gene has to be transcribed, translated, and make its way to the operon. The operon is always linked. We talked about linkage, right? The operon, mutations in the operon are always linked together. Mutations in the regulatory gene are unlinked. And we've used the term cis and trans before. That's going to be really important when we start using genetics as these operons. So this is an example of an operon in your book. So we're going to hopefully start getting familiar with these types of diagrams because they're what the book uses to try to communicate the phenotype. So that's what I want you to pay attention to. It's communicating two things. It's communicating the structure, so the DNA sequence that will tell us when there's a mutation, but the important part is the phenotype. So on the left-hand side, we have the promoter and the regulator. So it's making that regulator protein. So that protein can then interact with the operon, the operator. So if there's a mutation there, we would see it. And whether it looks different or whether it can if not bind to that operator. So in the operon on the right-hand side, promoter, operator, structural genes. Structural genes make proteins that do something. They're often catalyzing reactions in metabolism. Metabolism is like taking one chemical molecule and changing it to something else, right? So from lactose to glucose. So any questions on that? So I hand drew one. This is what the book looks like. The key point is there's those three really important parts of the opera. Structural genes, operator, promoter. And then somewhere else in the genome, regulatory gene, a regulator gene, promoter. So there's a lot of moving parts, but we can think about the consequences that they have on gene expression. So questions on that? This is not a specific gene. This is just like, we'll add a lot of specifics when we get to the laptop stuff. Okay, so we have this regulator gene. How is it actually interacting with the operator? What's the best way to describe these types of interactions? So currently we're getting answers like evenly spread. I don't think you guys can see the distribution, but it's equal representation. We have like 40 people answering. So does anyone want to defend an answer or eliminate? You can do that too. You guys think you're going to call my bluff? Not going to happen today. Yeah.
I guess like when I think of covalent bonds, I think of that being like more permanent and a lot of energy has to go into it, and like hydrogen bonds can be like formed like really fast and it doesn't require a lot of energy, it's kind of temporary.
Yeah, so that's what I was thinking. So if you didn't hear all of that, really, there's two answers, A and C are covalent bonds to different things. They're both covalent bonds. B is hydrogen bonds. Hydrogen bonds take a lot less energy to make, and they can be transient. So that's the logic I was thinking too. So I would say the best answer here is B. Further than that, covalent bonds between the sugars are the backbone of the DNA. So if you were trying to make a covalent bond, you'd have to break the DNA. That's not going to be good, right? If you're making and that's kind of A is the same thing. If you're making a covalent bond with DNA base, there's not many things to covalently bond with. You have all these hydrogens and polar nitrogens that are making hydrogen bonds. But if you wanted to make a covalent bond, you have to break something. So you don't want to break the DNA. You want to kind of just transiently interact with the DNA through these hydrogen bonds. So thank you. Does that make sense? So why is that important? Because the regulator protein is either there, it has to come off, or it's off, it has to come on. It has to kind of do this transiently. It can't always be stuck in this covalent bond for the DNA.
The other thing with that is hydrogen bonds are harder to break apart, so it's structurally more sound.
Covalent bonds are harder to break apart.
Okay.
They require, I guess it depends on how you define harder. They require more energy
Right.
to break apart. Hydrogen bonds require no energy for this electromagnetic interaction, not a physical. So how are genes regulated? Now we're going to get into a lot of kind of like definitions of how we're going to define genes and operons. And so what we need to think about is this is kind of from the perspective of the operon, but it's a product of the regulator. So we're going to start with this definition, then move to another, and then kind of combine them. So I think this is the best way to walk through it. Your book just kind of like jumps into it. So what do you think of if I say a gene is constitutively expressed? What does that bring to mind?
Continuity? Continual?
Yeah, that's what I think. So constitutive means all the time. So this would be a gene that's expressed all the time. These are kind of rare. I mean, there are some housekeeping genes that are expressed all the time. I still think they're regulated. Your book kind of says this is under normal cellular conditions, because it will try to then differentiate between mutants that have constitutive properties. So I'm not going to say under normal cellular circumstances. I'm just going to say it's expressed all the time. It's rare for a subset of genes, or when there's mutations in regulatory genes or in compounds. Okay, so what might be the difference between positive and negative? What does that kind of make you think of? Which one? Yeah. So positive control, the gene needs to be turned on. So it can activate or stimulate gene expression. So again, this is what's happening at the gene, but a product of the regulator. We're going to have positive regulation, negative regulation, but we're also going to add some other things. So negative would be the opposite, right? If you're inhibiting something. So there is feedback built in. Yeah, there's a comment in the chat. We will see feedback. It's really interesting in these bacterial systems, especially when you're thinking about gene regulation in terms of metabolism. It's often the metabolite that acts as a co-inducer or co-repressor. So there is feedback built in, but we haven't seen that yet. Okay, so there's three terms: constitutive, positive, negative. We're now going to combine them with these next terms: inducible and repressible. So inducible is expression is usually off and needs to get turned on. And then repressible is the opposite. So how are you going to induce something? You can do it through either negative regulation. You can't really do it through positive regulation. So it's not quite factorial. It's not that you can combine positive with both of these. Kind of negative inducible, negative repressible. And we'll see an example of this. So here's an example of an inducible operand. So again, this is kind of a general operand. It doesn't have any specifics about the metabolites. We can add specifics onto it, but what's the general principle? So something is going to change in the environment, right? If we're saying that the gene is normally off and needs to be turned on, there's going to be a switch. So that's what the black or the gray boxes is going to start showing in the diagrams in your book and in these slides. Like condition 1, so this is a condition no inducer present. Condition 2, inducer present. So inducer is a molecule that interacts with the regulator gene to induce, to turn on gene expression. So we have the promoter, the regulator, the active regulator protein is binding to the operator. This is a very specific type of inducible, so don't think of inducible as this, just think of the gene is off and then it's on. And so can you see the difference? This is happening through the release of that regulator protein, right? So right here, the regulator so off, on, due to that regulator piece. Nothing inherent about that operon. We would classify this operon as inducible if the regulation is coming from the regulator. So this is a repressible, right? So right here, the regulator is binding. So it was on, then off. So kind of the exact flip in the slide we just saw. So these terms are really important. Hopefully, seeing diagrams, you can kind of work backwards and figure it out. But if you know these terms moving forward, it really helps kind of set the stage of what you might expect when mutations occur. The mutations in regulator proteins are going to affect inducible versus repressible operons very differently. So we have kind of negative, positive, constitutive. We have inducible, repressible. We can only really combine. Positive is just positive. If the gene that's kind of off, you turn it on. Constitutive is always on. So you can't kind of mix those with inducible or repressible. The only prefix we can add to inducible and repressible is really negative. So hopefully you can kind of see the two. Negative was something's going to inhibit. Inducible means it needs to get turned on. Right? So it really is the combination of both of those terms we've been talking about. So the control at the operator is negative due to that regulator gene. That control at the operator has expression off, and then it'll get turned on. So this requires an inducer. So that'll be a small molecule that interacts with the regulator. We're going to see the same slides we saw, but now we can add more descriptive language to these operons by kind of combining these terms. So this is the exact same slide you saw that just said are inducible, but now we can say it's negative inducible. So the negative part comes from here. So the regulator inhibits expression. And kind of the before and after is the inducible part. Before, the gene is off, but after the inducer, the small molecule is present, the gene is turned on. So that's why we're using the two terms to describe the before and after state and describe the nature of that regulatory protein. Is that sort of clear? So the next couple of slides are going to be on repressible, negative repressible. Well, let's just think about it. What does anyone have a guess about what a negative repressible operon is going to look like? So negative, you're still going to get an inhibitor or the regulatory protein binding and inhibiting transcription. But the positive part, yeah. Yeah, it's going to flip the before and after. So it's going to be on, no regulator interacting with the operon, then after, direct inhibition of transcription. So we'll see that. So again, negative is meaning the control of the operon. The regulatory gene is going to inhibit. And repressible is it starts on and it turns off. And the small molecule here is called co-repressive. So I know these are a lot of terms and a lot of definitions, but I think they're really, really important. So we should practice using them very specifically to kind of build that habit. When you're faced with a problem, you immediately identify the patterns you would expect to see when you hear about a negative inducible operator or a positive operator. The one tricky thing is lots of operators can be both positive and negative for the regulator. So usually we'll start with like the negative, and then we can move to the positive. So this is the same slide we saw for repressible, but now we can add that further language. It's negative repressible. So here's the negative part. That regulator is binding to turn off transcription, and then this is the positive part, the before and after. It's on, then it's being turned off. So the off is happening due to negative regulation of the operator. But because it's on to begin with, it's repressible. Any questions on that? Does that sort of make sense? You wouldn't see positive repressible, right? A positive would be that the protein comes to stimulate translation or transcription, right? That wouldn't make sense in terms of repressible. It also wouldn't make sense in terms of inducible. So only these negative controls are added for repressible. So in positive control, the regulatory protein, you can call it something different to try to move away from the negative regulation. So to try to use a new term, we also often call it an activator. It may or may not need a co-activator, a small molecule to facilitate activation. So we will see an example of positive control bacteria. But this type of positive control is way more common in eukaryotic gene regulation. We have lots of transcription factors that are binding, right? So we don't talk about transcription factors in bacterial systems. We talk about kind of these regulatory systems. But I want to make this point now, so hopefully you can see where we're going when we get to eukaryotic cells. So eukaryotic cells are often positively controlled through transcription factors. So let's combine these things and ask a top-up question. So in a negative repressible operon, lots of words, the regulator protein is synthesized as a what? I threw a bunch of words at you. Now you have to think about how to combine this to answer this question. So we have currently a tie between C and D. So if you kind of know the terms already, you might just choose an answer, or you might be able to just eliminate A and B. I have to picture it in my head. So I'm going to draw stuff, but I want you to tell me what to draw. So what do you want me to draw first? You're making a mental image of what this looks like. What's your mental image have it? We need to differentiate between an active repressor and an inactive repressor. And we're going to try doing that by drawing something.
We want to draw the structure.
Sure, let's start with an opera. Sure. So what do I need in my opera?
Structural genes. And then a promoter right.
Sure, yeah. We need a promoter. We need an operator. We need some structure. Okay, good. How many operators should we draw? I would think two for this reason. Right? We want it before, after. So what does the operator look like before and after? What term in this question is telling you what's happening before and after? Yes, and what term helps you decide that?
Repressor.
Yeah, repressible, yes. So this is giving you the before and after. So you said before it was off, right? And after it was on, or is that backwards?
That's backwards.
I think so, I think I just misheard you. See, it, before it was on, and now it's off, right? Yeah, that's our definition of repressible. On, we're shutting it down. Okay, what else do you want to draw? What can we draw that gets at this part, negative? What is negative tell me?
That's promoter.
Yeah, I just can't hear you. Yeah, the operon is controlled in a negative fashion by the regulator. So what does the regulator do? Or where will we see the regulator? In before or after? So we have to have a little regulator. Oh, that's not that good. Like Wu-Tang Clan, right? There's our regulator. What is it doing before and after?
It's not doing anything before.
Yeah, if it's a negative regulator, it's gonna turn stuff off. So before, it's just hanging out. Here, actually right here on the operon, right? On the operator. So that's where we get negative. The regulator gene is negatively controlling the operator by physically binding to the operator and turning excitation off. So we've already ruled these two out, right? They're not activated. Because we're dealing with negative control, not positive control. We've drawn a little bit about repressible, on and off. We've drawn the negative, something happened. So what happened? So we're just missing this last term, active or inactive. So which is it? What was the original state, and is it active or inactive?
Inactive.
Yeah. So if we're thinking of activity, its job is to repress and negatively control. Here is inactive.
Well, once it
Here
binds, it
is
becomes
active
active.
due to the presence of a repressor, right? A co-repressor. So there was a conformational change in this protein. Here, there's no co-repressor. Here there is. They draw them as like those little small molecules that interact, right? The co-repressor activates the repressor, the regulatory gene, and allows it to have negative control over this operon. So the original state would be before. So the best answer here is B. Questions on that? I know that's a lot, but that's where we're going, right? We're going even past this. We're going to genetics controlling this. So it's really important that we're all on the same page now, or the genetics will not make sense. There's a couple of things in the chat. The regulator won't be binded while it's on. The regulator binds it off. So that occurs, I think, that would occur in the negative inducible. So an inducible operator that activates the regulator is first active, is on. Then the small molecule comes on. Or the opposite, you can have an inducer that deactivates this. So I think these terms are really specific to the operators of thinking of. In this case, I'm pretty sure it's inactive and then it's active. Operons are only in bacteria, so that's the second question. So here's the operon that we're gonna be spending the most time with, the last operon. So it's named, the last is the name of the gene, the structural gene. And they're involved in lactose metabolism. So genes in bacteria and fruit flies are often named for the mutant phenotype. So if you mutate genes in this operon, you cannot metabolize lactose. So lactose is the sugar. So it's a negative inducible operon. Involved in lactose metabolism. And the inducer is allolactose. So induction, right, inducible means you're off and you're getting turned on. Repressible is what we just saw in the previous example. So we have a promoter, we have an operator, we have some structural genes. So we call the operator has a very specific kind of genetic symbol. So we're going to use black O. That's going to be the operator. Black P is going to be our promoter. Comes the operon. That's really important. There will be another promoter, but we're not going to really think about mutations in that other promoter. We're going to think about mutations in the last operon promoter. We have our regulator gene. So we call this black I. So that's, the regulator. Sometimes your book calls them repressors. I think your book uses a lot of language interchangeably. They named it I, they found out it's an inducer. So there's a lot of terms kind of built into that genetic code that we have. And then there's three structural genes like X, Y, and Z. The identity of the structural genes is not super important, except for the fact that oftentimes the readout of the mutations is the presence of lactose, allolactose, and glucose. And that's because the structural genes kind of break those molecules down. So one of the structural genes codes for the enzyme beta-galactosidase. So it makes lactose into allolactose, the inducer. It also sends it down to the rest of the structural genes, like a permease and something else. So just a little bit on the structural. So ACE is an enzyme, it does something to sugars. Beta is referred, I think, to the conformation. But I don't remember 100%. Black Y is the permease. So again, I don't think it's really critical to remember all these genes except for the fact that they're a readout. On an exam, I'd probably give you like a rough diagram of the lac operon. I probably wouldn't give you like a full figure in the book, but enough that you can do genetics with. So this black Y, Z and Y is important for discussion next week because it'll give you two phenotypes. The presence, so you can use the small molecules to identify mutations, or you can use the presence of these proteins to identify mutations. So it's going to give you mutations in lots of different parts of the lac operon, but in lac Z or lac Y, so it's kind of like the one protein is being expressed, the other is not. How does that affect downstream metabolism of lactose? So that's why I wanted to be explicit with what these two genes are. So when you see the discussion and it's telling you whether permease or beta-galactosidase is present, you kind of make that connection that, okay, those are the two products of two different structural genes within that same operon. transacetylase, it's moving an acetyl group to somewhere else. So here is kind of the before picture, right? Or kind of the state without the inducer. So we have BLACI, the regulator gene on the left. It's making an active regulator, right? So this is the opposite of what we did, what we drew in the top half. It starts out active and actively binds to the DNA. The inducer is going to inactivate it to keep it off the DNA. So RNA polymerase can't get to the promoter. It's physically blocked by the promoter. Maybe if the promoter was bigger, it could like sit there and wait, but that's really inefficient for a bacterial cell. Why is it wanting a polymerase to wait around on an operon that's not accessible? So this is a figure from your book, the promoter. So this is what I kind of was alluding to. There's not a specific sequence of DNA between promoter and structural genes, and the lac operon is kind of a special case where the operator is actually the end of the promoter and the beginning of lac disease. So it doesn't, operator doesn't have to be unique DNA sequence in the sense of it takes up a physical space between structural genes and promoters, but it does have to be a unique DNA sequence in the sense of that regulator protein will recognize it and bind to it. Does that make sense? We've been drawing them as separate entities just to think about all the parts. So if you see this, don't be thrown off by the fact that there's not like an additional colored square representing the operator. The operator is the DNA kind of at the junction of those two other chunks of DNA. So when lactose is not present, the gene is off. We add lactose to the system now, and the gene is turned on. It's inducible. There was negative control. This is where we get negative control. And now it's on. So that's the inducible part. Off, then on. Negative control. That regulator was stopping transcription. So why do we care about lactose? Because lactose, so this is the other caveat, I guess. And we'll explain the caveat after. Lactose is in the system. It's, part of it is metabolized to allolactose, which is the co-inducer. So you can say inducer, co-inducer. It's going to inactivate the regulator. It can no longer bind to the DNA. RNA polymerase can come to that promoter. So you might be thinking, well, if the structural gene, so this is the product of last disease, if beta-galactosidase is required to metabolize lactose to the inducer, how did that ever happen? Because even though he used this as a model system to think about negative inducible genetics, it's never like all the way shut off. Once in a while, it's like blipped on in the absence of lactose to make beta-galactosidase. So that won't affect us moving forward. You can just think of it as binary, like off, on. But I know some of you will think, well, how was it ever turned on if beta-galactosidase is required? That's because it's never all the way off. So I don't, if that's confusing to you, just ignore everything I said. Just think of off, on. But if you're curious as to how that can happen, we will eventually see positive regulation through glucose. So those are the kinds of small molecules we take in. We often define the system by the presence of lactose and glucose, but remember it's really allolactose that is the inducer. And so this is just zooming in on the operator. So we haven't really talked about consensus sequences. I'm not that interested in them. But if you zoom in on kind of that repressor, you can see the DNA sequence. So it's kind of the very last couple of bases of that promoter. The promoter and bacteria are really small. And then it's the beginning of that structural gene. I think this is our last topic. So in the presence of allolactose, the lactose stressor is what? So you can just look at the previous slides, but the goal is to have you think about it. So how did we think about it last time? We drew two operons. As I was before. So what kind of regulation is the last operon under?
Repressed. Repressor.
Yeah?
Oh, negative.
So inducible means it has to be turned on. We have black eye hanging out over here, making our little regulator. So where is allolactose coming into this situation? So where should I draw the regulator, the product of black eye, in the before picture? Yeah, it should be sitting on the operator. That's the negative part, right? So again, before and after is the inducible part. This is the negative part. So it's hanging out, and then allolactose comes in, and what happens? It's no longer on the operator. It's inactive. The regulator protein is now inactive. So the best answer here is C. Right? It's just the same way saying it. The inactive regulator cannot bind to the operator. Transcription is turned on. So those are the key pieces, right? I didn't have to. The only thing I needed to answer this question was to know how the operator is regulated. Negative, inducible. The inducible got me my before and after picture. The negative got me that the regulator was physically attached to the DNA in the off conformation. So then, all that leaves is the allolactose. I don't even have to know allolactose is the inducer, but I know if it's present, it likely is the inducer. So now it's binding to the regulator is not binding to the operator. Does that make sense?
Oh, yeah. Okay.
So again, next week we're going to do a lot of genetics, but I think a lot of questions like this are fair game. For any hypothetical operator, all I need to tell you is negative inducible, negative repressible, positive, and you should be able to draw pictures like this. Maybe not right now, but you can think about it, think about what we've done. And that's how I like to approach these problems. So kind of how we had our algorithm for pedigrees, we have an algorithm for operators. What is negative? So draw two operators before and after. Identify whether it's inducible or repressible. That's going to tell you if it's off-on or the reciprocal. And we're almost always going to deal in the off-on situation with negative. The negative being the physical binding of that regulator to the operator. So positive wouldn't look like this, right? Positive is just going to be. Any questions on this? Okay, that's my last slide. I'm happy to take more questions. There's another reading homework due Monday that's related to this. So there's a small one due tomorrow, and then a big one due Monday.
Your highlight side too. Oh crap.